that you were going to see this again. So everybody make sure you're looking at page 102, because I don't have my transparency with me, I think because I had a lecture yesterday and forgot to put it back into this folder. So there are two nucleotides. The nucleotide at the top shows an S35 DATP. So if you need to incorporate radioactivity into a growing chain of DNA, this is how you do it. The phosphates are labeled as alpha, beta, and gamma phosphates. So if you're trying to use something like a phosphatase is going to take the end phosphate off of a nucleotide and put it on the 5' end of the DNA. So in that case, you would want to make sure that your gamma phosphate is the one that's radioactively labeled. And you can have companies, you can order specifics. You can order it, alpha labeled, you can order it gamma labeled, etc. So if you need your radioactivity to be incorporated into a growing chain, you're going to order it with a alpha label, because this is the alpha phosphate right here. And so this is the one that's going to get incorporated into the growing chain. And the only difference between this alpha S35 DATP and a regular DATP is the radioactivity. It's still the DNA molecule with the hydroxide down here. Now, in addition to this molecule, as I said in lecture, the dideoxynucleotide is very important in Sanger dideoxy sequencing. And so in Sanger dideoxy sequencing, you have those phosphates, but down here, you're missing this hydroxyl group right here. And because you're missing the hydroxyl group, if this base gets incorporated, whether a G, a T, or a C, if this base gets incorporated into a growing chain, nothing more can grow onto the end of this DNA right here. The chain will stop growing, and nothing else will happen there. So let's talk about what happens during sequencing. And you have this page just a couple pages later, also in your lab manuals. Just turn the page and see what I'm talking about. Okay, so to do Sanger dideoxy sequencing, what you need is you're going to have an unknown sequence, and you want to know the sequence of this, but next to this sequence, you need to have a known DNA sequence. In my lecture yesterday, you were able to find a known DNA sequence by adding adapters onto both ends of the unknown DNA sequence. But in this case, generally you're sequencing something that's maybe been put into a plasmid, and if it's in a plasmid, you know the sequence of the plasmid, for example. So you're going to have a known DNA sequence that's next to your area that you need to get sequenced, and because you know the sequence, you can make a primer that is going to bind to a specific sequence specific to that known DNA sequence. And you can design any primer that's going to fit that known sequence. And you anneal them together. And then what you do is you separate them into 4 different reactions. So this is the traditional Sanger sequencing, and I'm going to explain in a minute how it differs now, and you're going to see both ways. So you put it into 4 different reactions. You're going to do a G reaction with it, for example. And in a G reaction, you're going to have S35 DATP. So anytime there's an A that's incorporated by the polymerase, you're going to have a radioactive A added. And you're going to have DGTP, DTTP, DCTP, and a certain small amount of that dideoxy GTP. So a certain small amount, because you don't want every single G to stop with the first G. You want maybe 1% of the G's to stop and the rest to go on. So here's what, and again, this is in your lab manual, so you can follow along. So here's where the known, here's where the primer ends with that A right there. And then the very next A, you're incorporating some radioactivity right there. So now it's radioactively labeled, able to be seen on an X-ray film, for example. And then eventually you get to the first G being added in there. So it's a first G. There's a certain probability, because this is a G reaction, and there's some dideoxy G in there. So there's a certain small probability that it's going to be a dideoxy G. But most of the time, it's going to be a deoxy G that gets incorporated, and it will continue along. But it maybe in 1% of the time, it'll be a dideoxy, and the chain will stop growing right there. But again, most of the time, it continues. So then we get to the next G, which happens to be right next to it. And again, certain small probability that's going to end with that G. Most of the time, it's going to continue. And the same thing will play out every time there's a G that gets incorporated into the sequence. There's a certain probability that a dideoxy will get incorporated and the chain will stop growing, but most of the time it will keep growing. So if we had a film, if we put these on a film and ran the film, separating by size, in all the G lanes, and again, this is also in your lab manual. So we have all the different G lanes. So we have G, after including the primer, we have, G stopping here and here and here and here and here and here. So all those G's you saw on the first page are particular sizes, and you run them on a gel, and you're going to get all the different sizes of G's separated out. And then you also have an A reaction. And in the A reaction, it's the same thing. You also have a certain small probability every time there is an A that it's going to be a dideoxy A. The only difference for an A reaction is that first A still has to be radioactive. Because if you incorporate a dideoxy A before you get your first radioactive A, you're never going to see that on the gel. So it just means that you need to incorporate a radioactive A in there before you can see anything on the gel. But after you have that first radioactive A, every time there's an A incorporated, if it's not a dideoxy, it's going to be a radioactive A. And so every time there's an A incorporated, again, there's a certain probability that it will be a dideoxy A. But if it's not a dideoxy A, then it keeps going. So maybe 1% of them are going to be dideoxy and 99% continue right on. So you're going to have, every time there's an A, there's going to be a particular size of that. And again, you can run that on a gel. When you run it on the gel, you run the G's next to the A's. So this is our G lane, and this is our A lane with all of our different sizes for the A lane. And then you're doing this just the same way with the T's and the C's. So you have G's, A's, T's, and C's labeled and run in a gel, and they're separating by size. So in this traditional Sanger dideoxy method, you would read the gel from bottom to top, 5' to 3'. So remember, this is G, A, T, and C. So if it appears in this lane, you have a C. So you would read the sequence as C, T, A, C, G, G, A, etc., going up, 5' to 3', and you can get your entire sequence that way in this traditional film. So this was nice, but as people started sequencing more, they wanted to be more efficient. They said, how can we be more efficient with this? So what they started doing is, instead of using radioactivity, they started using fluorescent tags on the dideoxys. So they had fluorescent tags on the dideoxys. Let me pass this out. And because they had fluorescent tags on the dideoxys, this meant that everybody could run all the lanes in the exact same lane, because there was no more radioactivity. Each fluorescent, each nucleotide had its own different fluorescent color attached to it now. Okay. So let me turn the lights on and we'll talk about this. So if you were sequencing something with radioactivity, it took 4 different lanes to sequence with radioactivity versus just a single lane to sequence with this fluorescently tagged dideoxy lane. So you could get 4 times as much data from every single sample that you were running. So it was a much more efficient way of doing it. Okay, so what I want you to see is it's a chromatogram. So you have different little peaks up here in different little colors, and there's a sequence written above this first lane. So the sequence of interest, the sequence that goes with this first lane of chromatograms, is the sequence above it, not the sequence that's below it. So there's always a little bit of ends at the beginning when the program is like, well, we're not really sure if this is larger than this, but eventually it starts to resolve itself. So, for example, mine starts around base 22, C, C, T, A, C, T, 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 etc. So you can start reading that sequence as you go, and you can see that they are in 4 different colors. So they're all in the same lane, and they're run using capillary electrophoresis, basically the width of a human hair, a small sample of a jelly-like material that's inside of the capillary in capillary electrophoresis is kind of like acrylamide here, and it's allowing separation to occur as the different sizes are allowed to move through and have to separate based on the size that they are. So what we're going to do in the next lab, you're going to be taking your sample, and so there's 1 for each group. So I handed out 1 of these for each group. You will be doing them as a group. You're going to be having a single assignment with everyone's name on it in the group, and you're going to be submitting that. If you have 3 people in your group, you're going to be submitting the exact same thing 3 times onto Canvas. So make sure you have all 3 group members named on it, so that Teresa can make sure you're doing it correctly. So basically, let me get the other handout here. We have sequencing homework to do. I'll have one of these, I think, for everybody. Okay, so what we're going to be doing next time, you're going to get an X-ray film, and the X-ray film is going to have radioactive markings on it, much like that gel I just showed you how to read. It's going to have bands, and you're going to have to read the sequence from the X-ray film, and that's going to be what's called our wild type sequence. And what I've just given you, and there's different ones for the different groups, is a mutant sequence. So what you're going to need on there is, at the top or the left side, you'll see M number C number, like M2C6 or M3C4. Make sure you report that, and that tells Teresa what sequence to expect for your mutations. So what you're going to do next time is you're going to read the film to get the wild type sequence off of it, and you're going to compare that to the sequence that I've just given you here. And I'll be showing you on the film next time where to start reading and about where to stop reading. You'll read about 50 bases or so, and you'll read an area of this. And if you look at your DNA sequencing homework now, you're going to be sorry, too much talking today. You're going to be using the BLAST website to align the two sequences. So traditionally, as in the 1990s, for example, we had one person that would have to read the sequence off the film, be reading it out loud, somebody else would have a piece of paper and try to follow along. The alignment tools made life so much better. You could just, when you get your sequence, they would always give you a computer file telling you, here's the entire sequence that we've got, and then you had your known sequence that you started with, so that you were very able to easily confirm whether your mutations were present or not. You're going to have to do a little more work because I'm not giving you the computer file. You're going to have to look at it by eye. But you're going to put one sequence in the first space in the alignment box and the other sequence in the second, remembering which one is which. Doesn't matter which way you do it, but you have to remember which one is which. And it's going to show you an alignment of the sequences. So it'll look something like this. So where they agree, it'll give you a line matching them up. Where they don't agree, it'll give you a little blank space. There are no additions and there are no deletions. You may see some ends though. And when you're looking on this sequence right here, you might see an N in there. But then if you look below the N, you can probably see that, okay, well, I see different colors here. Maybe I see a blue and a green. Well, what happens is maybe the blue is overlaying the green a little bit, and so the computer algorithm is not smart enough to say, okay, well, I see the blue overlaying the green. They're a little bit too close for my program, and so I'm going to put N because I'm not really sure. So that's what happens with the computer. But you're smarter than a computer. So you can take it and you can look at it and say, well, I see that there's in the chromatogram, there's a blue peak quickly followed by a green peak. And blue is C and green is A, so the sequence would be CA instead of CNN because the computer can't really read the difference here. So you can use that to resolve some of the things. But again, keeping in mind there are no additions and there are no deletions. So what you're going to do is you're going to find the areas of mutation and you're going to report them. And there will be a total of 2 to 6 bases mutated. So there will not be less than 2, there will not be more than 6, and there will be in only 2 areas. So this is one area, for example, and you can see this, maybe it was originally a T and it got mutated to a C or vice versa. And so I'd say, okay, this is one mutation right here, and oh, now I have a couple bases down. This is, maybe I have 2 more mutations here. And you can show me the areas of mutation. Now, if you have, for example, only one base in between here, and maybe there's another mutation here, we'll call that an A right there. This is all one area. So this would all be one area, A, C, and then A right there, because it's only separated by one base. So if it's separated by one correct base, it's all one area. If it's separated by more than one correct base, then it becomes its own area. So you need to keep that in mind when you're telling me where the areas of mutation are. If you find more than 3 areas of mutation or more than 6 bases, you're going to reread the sequence to resolve discrepancies. If you read too far, you're going to find more mutations, which is why I'm going to be telling you where to start from on the film and where to end with on the film. And you're only going to read about 50 bases because that's where the mutations are that I'm interested in you finding. And so then you're going to do that, and you're going to find what chromosome it's on using the software. And then there's some questions that you need to answer: the difference between a processive and a distributive, and 3 enzymes which can be used to sequence DNA. And you're going to put that into a group effort. And so next time I will be showing you how to read the old style radioactive sequencing film, and you already have this to be able to compare it with, and you've got the explanation. Any questions?
So that would be Thursday?
That's what we're going to do on Thursday. You're going to read on Thursday. You're going to read the known gel for PCR, so that on Thursday you're going to be able to do your statistics and I'll be working with you on Thursday. You're going to be able to do your statistics and see how to achieve your goal there. And you're also going to be reading the sequencing film. So no actual lab work on Thursday, but it's going to be read this, read that, do the basic work day. Any questions?
So Kathy, you want me to run the band off or right at the bottom?
No, that's fine. Stop. Okay, if there's no questions, I'm taking off.